Soleno Therapeutics has announced promising results from their phase 3 clinical trial of DCCR. In today's presentation, you'll receive an update from Soleno Therapeutics on this trial. To begin the presentation, I would like to give you Anish Bhatnagar, CEO of Soleno Therapeutics.
Thank you, Susan. Good afternoon, everyone, and thank you for joining us today. I'm Anish Bhatnagar, CEO of Soleno Therapeutics. We're delighted to be sponsoring this conference and hosting this session today. Before we start, I'd like to first thank FPWR for giving us the opportunity to present today, and would also like to thank you, the PWS community, for your support over the last several months. As you all know, FPWR and PWSA USA led the petition asking for regulatory flexibility for DCCR, and it was unprecedented in its scale with more than 26,000 signatures. The virtual town halls that have been held by the PWS advocates, they underscore the stories of the caregivers and the subjects who've been in part of the clinical trials with DCCR, and they remind us that DCCR has the potential to be a very meaningful option for patients if approved. So thank you again. We've generated some really interesting data in the last several months, which we would like to share today. You've seen some of this before, some of it is new. The people presenting today will be those who have worked directly on the DCCR trials or have been instrumental in helping us characterize the data and better understand the effects of DCCR. So we'll start with Dr. Obrimba, who is one of our investigators in the 601-602 studies at Nationwide Children's. It's one of our high enrolling sites. Then Dr. Teresa Strong from FPWR will present the comparison of the long-term data with the Path for PWS study. Mindy Leffler will discuss the findings from interviews that were conducted with caregivers of more than 50 subjects in the 601-602 studies. And finally, Dr. Jennifer Miller, who's a PI at the University of Florida, will discuss the safety profile of DCCR. At the end of that, we'll take questions. And without further ado, Dr. Obrimba.
Okay, great. Thank you for that introduction. And as Dr. Bhatnagar mentioned, I've had the opportunity to treat many patients with DCCR over the course of the last couple of years. And I also want to disclose that I'm also a principal investigator for the LevoCare PWS study. Okay, perfect. So what is DCCR? So DCCR is diazoxide choline controlled release, and it's the tablet formulation of the choline salt of diazoxide. And it's formulated for gradual release from the gut over a 24-hour time period. And this allows for once daily dosing compared to the immediate release of diazoxide, which is dosed every 6 to 8 hours. And so this is a really important distinction because although the mechanism of action is the same, because DCCR is controlled release, it also allows for therapeutic doses that are 1/2 to 1/3 that of the immediate release. And so this is important not only for practicality of dosing, but also by potentially decreasing the risk for side effects or safety concerns. Okay, so what is the mechanism of action? So DCCR acts at a potassium ATP channel. And so it is a channel agonist, and these channels are found in many different tissues throughout the body, both centrally and peripherally. Most physicians are familiar with these potassium channels in the pancreas. In the pancreas, we have beta cells where DCCR acts to decrease insulin secretion and therefore reduces hyperinsulinemia. However, we know there are other direct effects of DCCR, and specifically DCCR acts to reduce neuropeptide Y, which is a hormone that regulates hyperphagia and thereby reducing hyperphagia. DCCR also acts in the peripherally, specifically on the fat cells or the adipocytes, by reducing fatty acid biosynthesis and decreasing fat mass. Okay, so this slide demonstrates the C601 and C602 clinical trial design. And so C601, or Destiny PWS, was a phase 3 multi-center randomized double-blind placebo-controlled trial in patients with PWS ages 4 and above. There were 29 sites participating, 20 in the US and 9 in the UK. And so 127 patients were randomized, and they were randomized 2-to-1 to receive DCCR versus placebo. Of those 127 patients, 120 completed a 13-week study, which included 7 study visits. And so these are the top line results for C601. These were initially released in June of 2020. And so the primary endpoint of the study was to address change from baseline in hyperphagia on the Hyperphagia Questionnaire for Clinical Trials at visit 7 or week 13 of the double-blinded treatment period. And so although you can see that those in the DCCR group did have a bigger decrease or change in hyperphagia compared to placebo, this unfortunately did not reach statistical significance. We did, however, see that 2 of 3 key secondary endpoints did reach statistical significance, those being clinical global impression of improvement at visit 7 assessed by the clinician, and the mean change or decrease in fat mass determined by DEXA. And so the C601 data that I just presented was analyzed through the spring and summer of 2020. And at that time, it was really hard to ignore the impact that COVID-19 and the pandemic was having on our daily lives, especially our children, and especially children with special needs that are so dependent on structure and routine. And so it was recognized that COVID was not only impacting our day-to-day, but it was also likely impacting the clinical research that we were conducting. And so that was supported by numerous publications. And we anticipated that these subjective endpoints, such as those that were being assessed in the C601 trial, were very likely to be impacted by these changes in daily routine. And so given these known impacts, the sponsor reanalyzed the C601 data, but now only including data that was generated through March 1st of 2020. And that was chosen prior to the assumption of any significant impact of COVID-19 or the lockdowns that proceeded. And so what we can see now is that the primary endpoint, the mean change from baseline in hyperphagia at week 13 or visit 7, now reaches statistical significance. So meaning those that were on DCCR had significantly greater change or decrease in hyperphagia score compared to those on placebo. And furthermore, now all 3 key secondary endpoints also reached statistically significant changes. And so now I'm going to review these results just in a bit more detail. And so this is a waterfall plot that demonstrates the hyperphagia score on hyperphagia, the HQCT, change from baseline through the reanalysis period. And so on the left of the y-axis, it represents change in hyperphagia score. A decrease in score, which would be desirable, would be represented by a bar below the median line. Patients that were randomized to DCCR are labeled in green, and patients randomized to placebo are labeled in blue. And so you can see that to the right of the graph, the majority of patients skewed to the right represented a bigger decrease in the hyperphagia score, and we would term those as our biggest responders. And those were more often green or randomized to DCCR versus placebo. This slide demonstrates the key secondary endpoint, the caregiver global impression of change. This question assessed the caregiver's subjective observation regarding global improvement in their child after 13 weeks. This used a 7-point scale in which caregivers were asked to rate their loved one as very much worse, no change, to very much better. And you can see that in the graph to the left, those that were on DCCR were much more often to be assessed as having improvement in global symptoms by caregivers than those on placebo. And to the right, you can see that twice as many patients on DCCR were rated as having improvement compared to placebo from their caregivers. This slide demonstrates the clinician global impression of improvement. So this question now assesses the clinician's subjective observation regarding clinical improvement using that same 7-point scale. And so this was really like in the eyes of the investigator. And so you have to remember that the clinicians, we were blinded. So meaning clinicians did not know if their patients were receiving DCCR or if they were in fact on placebo. And you can see, again, on the left, those on DCCR were much more often rated as having clinical improvement by the clinician versus those on placebo. And that was, by a comparison, 40% of those on DCCR were rated as having a clinical improvement from the clinician versus less than 7% on placebo. Okay, and finally, this is the 3rd key secondary endpoint. This assessed change in fat mass determined by DEXA over that 13-week period. And you can see that those on DCCR had significant decreases in fat mass when compared to placebo. Okay, so we did observe or assess additional endpoints that were examined in C601, and these included many behavioral endpoints. Specifically, we assessed changes on the on the PWS profile questionnaire and the developmental behavioral checklist. What we found is that we've observed significant improvements on most of those those core PWS behaviors that many of our kids are internally struggling with. So meaning we saw improvements in aggression, the anxiety, rigidity, and compulsivity. In addition, we also observed some of the improvement in some of those external or forward-facing behaviors. So meaning how our kids are able to communicate or socialize with one another. So we saw improvement in communication skills, interaction skills, and socialization on those treated with DCCR compared to placebo. Okay, and so why would we be seeing these improvements? Why is this happening? And so this slide is really demonstrating what is happening from a physiologic standpoint. So on the left, you can see this is insulin change from baseline. You can see that insulin is significantly lower over the course of the 13-week study in the DCCR group versus placebo. And so we really view this as a biomarker for the effect of DCCR, but it's really the downstream effects of insulin that likely is having the impact on all of these improvements in behavior and also from the metabolic standpoint. With this, we in turn also saw lower ghrelin. So ghrelin is that hunger hormone that we typically think of. We also saw lower leptin levels, which is typically produced by fat cells. And so this likely represents the decrease in fat mass that we observed over the course of the 13-week study. And then finally, we saw increased adiponectin. And so that is a good thing because adiponectin is a cardioprotective hormone. It may indicate improved cardiovascular health in those treated with DCCR versus placebo. Okay, and so that was the C601 data. And so now I want to show this slide again because I want to refresh your memory of the study protocol. And so following C601 completion, 115 of 120 patients elected to continue into C602, which was the open label extension study, meaning now all study participants were knowingly on study drug. If patients were randomized to placebo, they were now transitioned to DCCR, and patients on DCCR were continued on treatment. Currently, we have just under 100 patients that are continuing on study drug to this day, and many patients are now approaching year 3 of treatment with DCCR. Okay, so these tables show the demographic information for those patients that continued into C602. Patients ranged from age 4 all the way up to 44. And the mean age was around 13. So these were in most part still peripubertal, still growing children. The majority were treated with growth hormone, and at least 100 of these patients completed at least 1 year of treatment through the study. Okay, so what did we find after 1 year of treatment? So I think this slide very nicely demonstrates the change in hyperphagia that was observed over time. And so you can see that this is displaying the mean change in hyperphagia that occurred not just over the first 13 weeks of the study, but how we continued to see a decrease in hyperphagia scores throughout the first year of this open label extension. And so what we found after 1 year of treatment is that we observed a 50% reduction in the hyperphagia score from baseline to 1 year. So on average, at entry of the study, the average score being about 21 at entry, and after 1 year of treatment, you can see that the average hyperphagia score was just below 10. And so I think this observation really holds true to what many patients and families and investigators are seeing in real life, meaning the improvement in clinical symptoms and hyperphagia is not something that we see immediately. We see this often it can take a little bit more time. It's a gradual improvement over time. And one investigator stated the other day that treatment with this drug is not a sprint, but more of a marathon. And I like that analogy, and I'm going to continue to use that. Okay. So we again assess changes in behaviors following 1 year of study on DCCR treatment. Again, we can see significant improvements in all of those core PWS behaviors now across all behavioral domains. This slide shows that same data demonstrating the improvement in behavioral domains across the course of the study. And so you can see that in similarity to hyperphagia, we continue to see gradual improvements in these behaviors throughout the first year of the study. And so we see this in parallel to improvement with our hyperphagia scores. Okay, so we also examined body composition after 1 year of study. We found that while in C602, we actually did not see a change in body fat mass. We did see that body fat mass was at least unchanged, or it remained stable throughout the course of the study. And we recognize this could be for a couple of reasons. We have a growing population who is still growing and getting taller, getting larger. We studied this population during COVID. But what we did see is that we saw that lean body mass or muscle mass did decrease. And we also saw that lean body mass to fat mass ratio did decrease as well. And so this is what we are really excited about, because DCCR had a huge impact on the body composition of our patients after 1 year of treatment. And this finding, I think, again, this translates into some real world benefits. I have one patient that previous to DCCR would hike 1 to 2 miles each Saturday and Sunday. After a couple of years now on treatment, he's hiking 3 to 5 miles each day on the weekend. And he notes great improved exercise capacity. I think this is likely due to his improvement in lean muscle mass. Another patient that I've observed over the course of the couple of years, he's actually had to increase his caloric intake daily. And that is not something that we hear very often, right? And this is likely due to increased resting energy expenditure, meaning that as we all increase our muscle mass, we naturally increase our resting metabolic rate. Okay. And so again, we examined the same metabolic profiles in C602 after 1 year of study on DCCR. Again, we saw similar improvements in leptin and adiponectin. Not surprisingly, we saw decreased fasting insulin. And I think many people may worry, does this lead to high glucose? And this does not. However, because of these other favorable metabolic changes, we saw a decrease in something called HOMA-IR. And so this is a scientific way to measure insulin resistance. And so we saw decreased insulin resistance, which in turn means increase in insulin sensitivity. And so this is a good thing. And so even though we saw insulin levels were lower, our patients were more sensitive to the effects of insulin. And suffice to say, again, evidence of improvement in the metabolic health of our patients with PWS that were treated with DCCR. And so I want to finish up with some quick safety comments on safety. We now have over 100 patients that have been treated for more than 1 year. We have found this drug to be very safe. The safety profile is very similar to what we already know about diazoxide. The most common adverse events were as expected, that those being hypertrichosis or excess hair growth, some peripheral edema and swelling, and hyperglycemia. Although most of these events were very mild and self-limiting, and we saw no serious unexpected or adverse events. And so in summary, the results of the C602 data at 1 year show highly statistically significant improvement across all behavioral and endocrine and metabolic endpoints. And now we have years of experience with this drug, and it has proven to be very safe and effective. And so while we realize that this data is very compelling in terms of efficacy and safety, we also realize that in any open label study where all participants are receiving study drug, it's important to note the risk for introduction of a hope bias, meaning we would all be ecstatic and very hopeful that this new drug is being beneficial to our loved ones with PWS. And so it's important, we recognize, to continue to compare our data, our study population with external controls. And so Dr. Strong is going to present the C602 data compared to controls using the natural history study or the Path for PWS study.
As Dr. Obrenbaugh said, I'm going to talk with you about the comparison of the 601 and 602 studies of DCCR and PWS with the Path for PWS natural history study. So Dr. Obrenbaugh has introduced the 601 and 602 studies. For those of you who are not familiar with Path for PWS, this is a 4-year prospective study. It's non-interventional and observational. And it is meant to advance the understanding of serious medical events in PWS and also evaluate how PWS-related behaviors change over time. Some of you may remember that this study was actually initiated by the company Zafgen, which was developing drugs and thought that having this natural history data would be really important as a comparison. And we agreed, so we were working with them on that. Unfortunately, their drug didn't advance and they pulled out of the space. And FPWR, recognizing how important this data is, has continued with the study with the hopes that it might support drug development, other drug development. So the study is conducted through the Global PWS Registry, and it is parent-reported past medical history, as well as every 6 months, our caregivers go in and they complete height and weight. They talk about what medications their kids are on, and they do the behavioral assessments. And importantly, the behavioral assessments are very similar to those, or identical to those that are done in the 601 and 602 studies. In addition, we ask parents to report any serious medical events or thrombotic events that have occurred, so we can understand what is the rate of serious medical events in the population in general, as a comparison for drug trials. Next slide. So the importance of natural history studies. So for a long time, for several years now, the FDA has emphasized the importance of natural history studies, particularly in rare disease. A natural history study is a study. It's traditionally identified as the course the disease takes in the absence of an intervention. So it tracks how the disease or disorder changes over time. And it includes participants who are receiving the current standard of care, which we believe is the case for our Path for PWS participants. And it provides a population that is similar to the population that is enrolling in clinical trials, and provides an opportunity to compare endpoints in the natural history study compared to any ongoing clinical trials. So Path for PWS was initiated in late 2018, so it has actually run pretty contemporaneously with the Salinas study. The initial target recruitment was 500 individuals with PWS, but we were really excited that our community has been so supportive. We had more than 700 recruited and 650 who enrolled and have remained active. So that is the largest PWS study ever conducted, and it gives us a wealth of data to really understand how PWS normally progresses, and again, able to use that information to compare to drug studies. The study is mainly US participants, about 90%, but we also have participants from Canada, Australia, and New Zealand. And you can see the age distribution broken down. We really tried to pull in some additional adults with PWS, so about 40% are over the age of 18. The study team members are shown there. Dr. Miller and Dr. Sean McCandless provide guidance and medical oversight, and Lisa Matesevec is our PATH study coordinator. So Dr. Obrinbaugh has talked quite a bit about the 601 and 602 studies, so I won't to more detail about that, except the 601 is the initial randomized control portion, and the 602 is the open label extension. So how was this comparison of that data to the Path for PWS study done? So an outside company was used to compare the two. So we included participants from PATH who met the criteria for inclusion into Salinas study. So they had to be within a certain weight, they had to have a certain HQCT score, they had to be not enrolled in any other clinical trial, and they also had to complete the HQCT and other questionnaires at the same intervals that was being completed in the open label extension study. When we applied all those parameters, it resulted in a cohort and a comparison of 114 individuals from the 601-602 study with 229 participants in the Path for PWS study. And again, the analysis was done by an outside contract research organization looking at HQCT, change in HQCT scores, the PWS profile, and the comparison was done at 26 and 52 weeks, so 6 months and a year. So this shows you the demographics. Again, when we apply those parameters to narrow down the populations, we ended up with 229 from Path for PWS. You can see overall, it's pretty similar populations. The Path for PWS group is a little bit of an older population. The other breakdowns are pretty well matched. There are a few, a lower percent of individuals on growth hormone in Path for PWS, which likely is the result of the fact that it is more difficult for our kids or young adults to get growth hormone therapy after they reach the age of 18. So here is the comparison of Path for PWS HQCT scores, the change from baseline to 26 weeks or 52 weeks. And you can see in blue is the Path for PWS data. And in green is the individuals who were on DCCR. So as you can see for path for PWS, there is a little bit of decrease over that time. And if we look at the individual, like the waterfall plot that Dr. Obrimba showed for the path participants, we see some go up and some go down. So this is just what we would consider the normal variability of the HQCT. But as you can see, for those participants in the that are taking DCCR, they have a significantly higher decrease in their HQCT score. So they are much improved compared to the path for PWS participants. And, you know, as I said, there are some differences between the bigger populations, and so propensity controlling was done. So basically trying to match participants based on things like their growth hormone status and their age and their HQCT scores. So when those rules are applied, we see a very similar results. Again, significantly decreased HQCT score in the DCCR participants compared to the path for PWS natural history study. If we look at percent of individuals who responded using a cutoff of a decrease of more than 7 points of HQCT, we see that in the path for PWS study, about 20% of individuals had a decrease along those lines, but in the 601 and 602 study, those receiving DCCR, it was a much higher percent. So 60% of individuals had a significant decrease in that HQCT score. So this is just another way of looking at that data. We also looked at the PWS profile, which again is included in the path for PWS study. And you can see in blue, the PWS profile change after a year was very minor in our path for PWS participants. So the aggressive behaviors, anxiety, rigidity, etc., really stayed about the same over that year. And you can see the big difference in those individuals who were receiving DCCR in comparison, and that was significant across the board. So the conclusions here are that compared to the path for PWS participants, individuals who were treated with DCCR showed highly significant improvements in their hyperphagia scores as measured by HQCT, as well as significantly greater improvements in many of the behavioral features that are most challenging for families, including aggression, anxiety, irritability, and depression. So why is this so important? Well, you know, obviously, in a rare disease in particular, all data points are really important, and taking into account the natural history of the disorder, what would happen if we don't have a treatment compared to what happens if we do have a treatment, is a really critical piece. It provides evidence of long-term clinically meaningful improvements in hyperphagia with DCCR treatments, and it also provides evidence of long-term improvements in a number of the most troubling PWS behaviors that families face. And that is compared to the natural history of the disorder. So I just really want to thank every family who participated in the clinical trial, as well as every family who has completed these surveys in the registry every 6 months. Your data is so important, and we really appreciate your contributions to moving this entire thing forward, as well as the investigators for both PATH and the Destiny PWS study. So I will now turn it over to Mindy Leffler, who's going to some of the qualitative changes that we're seeing during this study.
Thanks so much, Teresa. I'm going to go over the results of the qualitative interviews that were conducted as part of a sub-study to the Selena study. And just briefly, some background on our research organization at Casimir. Our story is a personal one, as this is a list of studies that have failed to reach significance on their primary endpoint conducted in Duchenne muscular dystrophy. My son's study is in the left-hand column. And each one of these studies represents kind of blood, sweat, and tears of a large number of families that participated. And so that's really our mission as a research organization, is to ensure that no other rare disease families are left in this position of contributing their lives to study participation and ending up without answers. And we've since worked in over 20 diseases, both rare and non, to capture data that's reflective of the caregiver and patient experience in a rigorous way. So when we got involved in this project, our first task was to really start considering the existing functional outcomes in Prader-Willi and to take a look at the ways in which they may not fully reflect the experiences of patients. And we identified a couple of points that we knew were going to be problematic. The first two really impact the questions that are asked, on some of the questionnaires assessing Prader-Willi. And number one, just the inherent difference and the heterogeneity of the patient population. In our initial conversations with families affected by Prader-Willi, we had everything from families whose children were able to manage their own caloric intake to families, whose children could not be left alone even for a minute. So a really drastic spectrum of different types of patients. And in addition to that, you have the layer of food security procedures that are really specific to every family. So every family had kind of their own way of working around the disease, and also just the changes in standards of care in Prader-Willi, with this food security focus being more recent and impacting kind of the younger generation of patients, and not as much the patients that were raised before those food security procedures were put in place. So you can imagine that those 2 points really do affect the questions that everybody's going to find sensitive, if that makes sense. They're going to affect, the difficulty of coming up with a single question that'll be responsive to all different types of families. And then on the last bullet, this really addresses the impact of what's going on in the immediate situation for families and their response to questionnaires. So you can imagine, that if you're answering a question that asks you how often does a particular hyperphagic behavior appear, and you just have to say, well, this is how often it shows up, but you don't get any place to note on the questionnaire that, this might be a questionnaire that you're answering around the holidays, and you're going to a lot of, potential get-togethers with family members that have buffets around. You don't get to enter that detail. You just have to answer how often a particular behavior is occurring. And so you really miss that impactful immediate context around the answers that caregivers give. So that's why we implemented the interviews as a sub-study into this, into the Selena study. It does follow FDA guidance. They have patient-focused drug development guidance that they've put together, and conducting these types of interviews is in alignment with that guidance. It allows for the capture of information in a really patient-specific way. So, how do you know that things are changing with your child if you're indicating that they have? And then you can dig into those details, and then you can also ask about the impact of those changes and the meaningfulness of change in a real way, rather than kind of asking patients, outside of a study to hypothesize about what changes might be meaningful. This allows a chance to actually ask those families that experience the change about that impact. So in terms of the study design, there were 4 interviews that were conducted. There was one that was conducted at baseline, so prior to randomization into the DCCR treatment arm or the placebo arm. We did an interview at the end of the double-blind portion of the study, and then 2 interviews were conducted over the open label period of the study. One right at 3 months to kind of coincide with the duration of the double-blind, and then one at a later period to look at longer-term changes. The interviews covered 5 different sections. We talked about kind of background, and then we focused on food-related behavior, more generalized behavior, impact on daily life, and then we concluded. If a change was discussed, the caregiver was asked to indicate whether it was a positive change or a negative change, whether it had a major or minor impact on their child's quality of life, and whether that change was meaningful to the family, and to describe the meaning of that change. The interviews were all recorded and then transcribed, and then we had trained research scientists that conducted the analysis and kind of analyzed the transcripts for different themes. And what we found is exactly what we expected when we looked at the baseline interviews, which is the heterogeneity that everyone has always spoken about with Prader-Willi. We absolutely saw that in this study population. In food behavior, we had parents that were talking about their children as not really being as much hyperphagic, as rigid about meal timing, all the way up to someone who was extremely driven by food seeking. Behavior-wise, we had people that were able to self-regulate and had a lot of self-awareness, all the way on the other end of the spectrum, with patients that lacked that ability almost entirely. And then same thing in daily life, awareness of behaviors, definitely aware but couldn't help themselves, all the way up to someone saying that they had no self-awareness that there were any issues whatsoever. So the baseline interview data just underscored how different every person with Prader-Willi is from every other person, which kind of highlighted the reason behind conducting these interviews in the first place. So in the interviews at the end of C601, this really does tell the same story as the data that Dr. Brumba covered, which is that when we looked at the end of the interviews from the end of the randomized portion of the study, we had a higher relative number of caregivers of patients on DCCR describing major improvement than we did of caregivers of patients that were on placebo. And then when we took a look at interviews that were just conducted prior to March 1st, that difference got even more marked. So the difference between the description, the number of patients relatively that were on DCCR that were describing major improvements, was much more, there was much more difference in those numbers. And so because we have these qualitative descriptions, we have these really rich descriptions of what was going on in people's lives at the time and why. And so we were able to dig into the impact of COVID. And we had families that said things like this quote, in this last 2 weeks, it's been much, much worse. She will really scream and hit at the drop of a dime. It comes out of nowhere. Again, it's really hard now because she's frustrated to no end with quarantine. So that has thrown a really unique wrench into everything. And then conversely, we had people that said things like, I can't imagine where we would have been in lockdown and transition to high school to class if we hadn't been on this medicine. I truly believe that this medicine has helped cope in a way and given him tools to be less anxious. So the reason why the introduction of COVID was so difficult for the results of this particular study is that it's not as though every family was impacted by COVID in the same way. It was that just like Prader-Willi itself, every family was impacted in different ways. So it became really difficult to interpret the data, post-COVID, because for some people, kids were home all the time, and so they weren't out in situations where they would notice more hyperphagic behavior outside of their food secure home environment. And in some families, it got much, much more difficult, with the quarantine. So it really did highlight why COVID had such an impact on the data in the study, and the fact that it was so truly different from one family to the next. So in the long-term interview analyses, it was very similar. We focused on change from baseline to after more than 1 year. And we included evaluation of the same types of parameters, looking at caregiver perception of change in those 5 kind of stratifications of change. And we used descriptive statistics to conduct the analysis. And the results on that were also very interesting and very similar to what Dr. Brumba covered in her data set. It tells the same story. So if you just look at the solid green, those are the families that described very meaningful positive change. And then the dotted green is patients that just talked about positive change, that was a lesser degree than very meaningful change. So what you see is, large numbers of patients, of caregivers' families, describing positive change across these different topics of aspects of life that are affected by Prader-Willi, from 47%, all the way down to 41, but large segments of the population describing positive indications of what was happening. And then, I've been asked about the impact of these conversations with families. And what I would say is that when we conduct these types of qualitative interviews, when I hear something that sounds more like a placebo effect, it tends to be more general descriptions of patient behavior. But what we heard in contrast in these interviews were very, very specific, patient-specific examples of change that was occurring. These are caregivers that were watching their children closely, knew exactly what to look for in terms of each family's kind of pain point. and noticed change within that. So, you know, a patient that makes his own lunch, you know, and cooks with them, they don't have to keep things out of reach anymore. They don't have to lock things anymore. Families that were noticing patients eating slower than they used to. This was a really common one, people talking about different behavior when they go out to eat. And families that, you know, when they would go out to eat prior, they would have, you know, tantrums to the point of taking people out of the restaurant, but now not having any issues going out to eat. So again, you can understand why once quarantine started, the data really got turned on its head because there weren't opportunities to go out to eat and put patients in those more stressful situations that would test their behavior. To families that were, you know, just commenting on, you know, the lack of kind of constant inquiry about hunger and food. So this slide just kind of talks about this really patient-specific ways that families were noticing change that speaks to the heterogeneity of the population. In addition to the food-related behaviors, we saw similar patient-specific descriptions in non-food-related behaviors. So a lot of comments about anxiety, about increased calmness, more even emotions, and less aggression. You know, families that were noting less outbursts, and even some lessening of potential violent behaviors, and then less rigidity. So families that were talking about, you know, going on vacation and, you know, having a child stay on the beach until, you know, past dinner time and not even commenting about the fact that dinner had been missed. So again, lots of patient-specific descriptions in non-food related behaviors. So that's kind of a brief summary. I want to save time for Dr. Miller, but just from our perspective, you know, a couple really important thank yous to the families that were willing to share their experience. You know, we realize we're really lucky to be able to speak to you, and hopefully this was a nice opportunity to be able to tell us what you thought mattered. And obviously to the PIs that participated in the study that recognize the need to capture this data and be able to analyze it systematically, and obviously to Soleno for really making this investment and valuing the contribution that families can make to research. Thanks very much. I'll hand it over to Dr. Miller.
Thank you, Mindy. I'm going to just apologize now. I'm in the airport, so if people are talking over me, I apologize. I'm going to talk briefly about the safety profile of DCCR. As you heard from Dr. Obrimba, DCCR has a very well understood safety profile. Its parent molecule diazoxide has been used in Prader-Willi, or not in Prader-Willi syndrome, excuse me, in endocrinology for many, many, many years. And when I started this study several years ago, I had come off of doing multiple drug studies, and my biggest concern for any study in this population was obviously safety. And so I agreed to do this study without knowing whether or not the drug would have all the beneficial effects that we ended up seeing, but because we knew that the safety profile was really well understood, and I felt very comfortable with it. So this is a summary of the C601 safety data from DCCR versus placebo. And as you can see, there were multiple adverse events that were seen both in patients on DCCR as well as those that were on placebo. The top 3 adverse events that were seen in DCCR included hypertrichosis, as Dr. Obrimba mentioned, which is increased fine downy body hair over the body, peripheral edema, which is swelling in the lower extremities, and high blood sugars. Again, these are adverse events that are known well in diazoxide, the parent molecule for DCCR, so they were not at all unexpected in this population. What was nice to see and reassuring to see was that we didn't see any adverse events in the Prader-Willi population that were unknown with the parent molecule DCCR in other populations. So that was very reassuring as to the safety profile of the drug. You can see that with these adverse events, most, as Dr. Oberman, were very mild, grade 1 or 2. And you can even see that some of the placebo patients actually had some of these same adverse events as well. This slide shows you the change in fasting blood sugar as well as the change in hemoglobin A1C during the C601 trial. As Dr. Obrimba mentioned, because DCCR works on insulin levels, we expect some degree of higher blood sugars. And you can see that there was a very mild increase in fasting blood sugars on C601, as well as a very mild increase in the hemoglobin A1C. Hemoglobin A1C is an average measure of blood sugar over the past 3 months. these differences that we're seeing were actually not clinically significant. However, some patients on DCCR did have to go down on their DCCR dose due to a little bit higher blood sugars, and 4 had to start new anti-diabetic medications like metformin, which is an insulin sensitizer, which works well in concert with DCCR. 1 individual on placebo had to also start metformin to help with hyperglycemia as well. Additionally, 1 patient on DCCR had to start antidiuretic therapy, which is what allows you to pee out extra because of the increased edema or swelling. This is now the C602 1-year data analysis for the safety, and you can see here that as mentioned in C601, the adverse events were virtually identical. There was no difference in adverse events, with the top 3 again being exactly what we would expect: hypertrichosis or increased body hair, peripheral edema, and higher blood sugars. And what is really reassuring about these 1-year data is that you can see that all of these adverse events were grade 1 or 2. There was no grade 3 or 4 events at all in the 1-year data. This data shows the fasting blood glucose with the C601 and C602 data. And as you can see, even though there was that mild trend upwards in glucose in the C601 trial, it actually returns closer to normal by the 1 year on C602. And the same is true for the hemoglobin A1C. So in the DCCR group on 602, there was 9 patients that did have to start anti-diabetic medications to help with higher blood sugars, and no patients actually had to start diuretics because there was not as much peripheral edema seen over the course of C602. So in summary, the safety profile of DCCR was consistent with exactly what we knew the safety profile of its parent molecule diazoxide to be, and was consistent with prior experience with this drug in the Prader-Willi population. The most common adverse events were, as I mentioned, hypertrichosis, peripheral edema, and hyperglycemia. Most events were grade 1, and as Dr. Obrimba has already mentioned, there were no grade 4 events or higher, sorry, and there were no unexpected severe adverse events. In our experience at University of Florida, we saw that the hypertrichosis actually does start to recede over time, and most patients actually did not have hypertrichosis remaining by 1 year of treatment on DCCR. Peripheral edema actually was much more common in the adult patient population than in children, and the hyperglycemia, as I mentioned, does improve over time on DCCR. Interestingly, in typical patients who have high blood sugars, they actually typically lose weight because they're peeing out extra glucose. And patients with Prader-Willi, when they have high blood sugars, they actually gain weight, likely because they're peeing out glucose and so are a little bit more hungry, so they eat more. And we saw the same trend in people with Prader-Willi on DCCR with higher blood sugars. But when the DCCR dose was lowered or they started anti-diabetic medications, the blood glucose levels came down, as did their weight, which was, again, very reassuring. So we want to say thank you to all the participants and their families who gave up an extraordinary amount of time to participate in this study. And thank you to all of the institutions that were sites for this study. And lastly, the patients really wanted to make sure that this opportunity did not go by for them to express gratitude to Soleno for everything that they've done for the Prader-Willi community. And so this is not part of Soleno's presentation. They didn't even know that this was happening. So I apologize a little bit, but the patients did want to give this message to Soleno. So this is from the patients to Soleno.
For giving me hope. Thank you, Selena, for all your researching. I appreciate you. Thank you, Selena.
Thank you, Soleno. Soleno, for everything that you have done for me and my family. I can do a lot of stuff on my own now.
Thank you, Selena. Thank you, Selena, for DCCR. Thank you, Selena. Thank you, Selena, for DCCR. It's helping me and not eating food. I'm not like eating food anymore. Thank you, Selena, for the challenge that I've been on. It has helped me live a better life.
Thank you, Soleno, for all that you have done for the PWS community and with DCCR. We appreciate you.
Thank you, Dr. Miller. That was a surprise.
I have to go get on a plane now. Sorry.
Thank you for taking the time. I think, Susan, we have some time for questions.
Yes, take as much time as you need.
All right. So if I can ask the panelists to turn their videos and audios on. So there's a few questions here. Please feel free to type them into the Q&A. The first one is, when do you expect these results to be published? So the results are published in abstract form in a couple of places, but we are working on the formal publication, hopefully in the near future. As you know, it's kind of a process, and with 29 sites, we need a fair bit of feedback to incorporate before we can publish, but it'll happen. The second question is, in an October 2020 LEVO presentation, LEVO indicated that the FDA agreed to exclude data after March 1 in their primary efficacy analysis and ultimate NDA submission. Has Soleno also had that conversation with the FDA? And if so, did the FDA agree with that approach? As we have indicated in the past, we are in a conversation with the FDA at this time. We have been submitting additional data to them. And there's not much I can add to that, but to say that we are in the process of submitting the data that you saw today to them as well. And we're hoping that we'll be able to get to some resolution with them on what might be needed for NDA submission. The next one, I think, Dr. Obrimba, this one is for you. With the path for PWS data, is the thought that, actually for Teresa as well, is the thought that the longer a patient is on DCCR, the differences between those on DCCR and those not will continue to grow greater, or is the thought that the differences will end up plateauing?
Sure. Yeah, that is a great question. I mean, to this point, we have data up into the 1-year mark, but we're continuing to follow these patients through now years 2 and 3 of study, and we're continuing to generate information on hyperphagia scores. And so I guess time will tell and the data will speak for itself. Dr. Strong.
I would agree with that. The path for PWS study continues, and we've just started looking at the data, how things change over time, and I think we'll just wait and see.
If I could just add to that really quickly, just based on the qualitative results, one kind of interesting aspect that we saw was that patients did speak about change in a very patient-specific way in earlier interviews, and I think that there is a bit of a chicken and egg there where families have to be willing to put their children in situations that kind of stress test the hyperphagic behavior. And so there may be an extended period of time before they see enough change in that patient-specific way to feel comfortable kind of putting them in a situation where they might see changes that would then be reflected in more generalized questions like the HQCT has. So I think there's an aspect of that dynamic at play in the difference between the short-term and the long-term results as well.
I agree, and I think the fact that both the HQCT and the PWS profile capturing some of the sort of peripheral behaviors will be helpful in kind of giving a little bit more context to those changes.
Dr. Obrimba, I think this will be for you, since Dr. Miller is not here. What did you do to treat hypertrichosis in the affected patient?
Yeah, so we absolutely did see hypertrichosis in our females and our males. For the most part, the excessive hair growth of the hypertrichosis was pretty mild. Many individuals felt like it was mild enough to where they did not need to do anything specific to remove it. If it was more significant, some of our girls might have shaved some of their sideburns, and the boys really liked the extra hair. But for the most part, it wasn't significant enough to do anything out of the ordinary.
Thank you. Does a child have to be in a certain hyperphagic stage to get DCCR? So at this point in time, there are no trials that are enrolling new patients on DCCR, but in the trials that we have conducted, the patients did need to have what's called moderate to severe hyperphagia. Eventually, it depends on if and when the drug is approved, what the label would say. So that's what it would be. That's what would determine who would get DCCR. Can you explain what is included in the slide clinical improvements? Dr. Rimba, would you like to take a shot at that?
Sure. Should I refer to a specific slide or?
I think that the question is about what is caregiver global impression of change and clinical global impression.
Oh, sure, absolutely. Thank you for clarifying. So there is a caregiver global impression of improvement and a clinician impression. And so it's really essentially just asking the caregivers a one-liner to say, at this point in time, how would you rate your PWS loved one's symptoms of PWS? Do you feel like they are better from the initiation of the study? Do you feel like they're much better? Do you feel like they're the same, or do you feel like they're worse? And so it's just a very non-specific but global, sort of all-entailing question, asking for the caregiver or the clinician to rate their subjective opinion as to how that individual is doing in general.
Just one thing to add, the clinical global impression of improvement typically involves a physician bringing in more inputs. So, they can look at, they can ask other people as well, not just the caregivers. They could potentially ask others as well to give a more objective assessment, if you will. Are you planning to seek marketing authorization in the EU as well? The answer is yes. The timing is unknown at this time. Can you correlate from the interviews and the HQCT score a threshold which represents a significant clinical response as represented by HQCT? So Mindy, let me answer it and then I'll let you speak as well. So the responder slide that Dr. Strong showed, which showed a change of -7 as a clinically meaningful response, comes from an extensive analysis of the 601 data set. So we had an external group analyze the data extensively, look at different thresholds for response, and arrived at a change of at least 7 points being clinically meaningful. So Mindy, I'll let you address how you would correlate that with the interviews.
Sure. Yeah. So I think that there are definite correlations between those. And when you look at kind of large amounts of data, you see trends. But then there's the influence of individual circumstance into any kind of lack of correlation on an individual patient basis. So for example, if the HQCT is asking about something like frequency of a behavior, and there is kind of something going on with the family that's impacting the frequency, that's going to throw that potential for correlation off, for example. And then the impact of food security procedures that a family, raises a child under or lack of is definitely going to impact whether they're sensitive to a particular question. So for example, a family that's locked the garbage since the child was very, very, very young is probably going to have a child that doesn't try to break into the garbage as much as a household in which, that might not necessarily be the case. So I think there are some on a study population level, that correlation was seen. But then, when you drill down to an individual patient level, in rare disease, the devil is in those details sometimes. So.
Thank you. There's a couple of questions about plans for studies in younger children. Is there a rationale to pursue that? There is some interesting rationale. The changes in leptin and insulin that we're seeing in these studies are very significant. And there's published data that shows that there are certain times in the progression of PWS where you see spikes in levels of leptin and insulin. And there's a belief that if you can intervene at the time, that may be something of meaning. So we continue to research that. And yes, there is interest in moving that forward. Teresa, this is for you. How can the PWS community continue to help Soleno and advocate for the FDA to review an NDA?
Well, we do have a session tomorrow on advocacy and effective advocacy. I think that, the community needs to remain engaged. We need to let Soleno, sort of have that dialogue with the FDA and sort of see where that goes. But I think, being able to express what is meaningful to families, what the impact, of a drug like DCCR is, those are the things, those are the places that our community is going to be most effective. And I think that there, hopefully will be a lot of opportunity for us to be able to express that to the FDA in the coming weeks or months.
Thank you. There's another question about, is DCCR something that can prevent symptoms? as I said, I think there is some interesting rationale for, prevention of progression of PWS or an early intervention type of approach in PWS, something that we continue to think about. Will there be a maximum upper limit on DCCR, or will it be available without fear of aging out? So the study that we conducted was 4 years and up. So theoretically, we've enrolled patients as old as 44 years old. So when the FDA decides whether to approve a drug or not, they also look at the data that's been generated and they look at the age range. So typically approvals, if something is approved, are based on that age range. So our expectation is that if the data from the 601, 602 studies is used, then this would be available to adults as well. DCCR has some variable effectiveness. Some are great, some Levo is the same. So how do we sort this out? Who needs what? I am not sure I can answer that question. Dr. Brimba, if you want to try, feel free.
Not sure that I can either. It is an exciting time in which we have several drug studies showing promise. So that is a $1 million question.
Yeah, I think it's fair to say that this is a complex disease. There's many aspects to the disease, and so many diseases need to be treated targeting different mechanisms of the pathophysiology. So it would not be a surprise if you're looking at more than one drug eventually to treat subjects with PWS. Teresa, did you want to add?
Yeah, I did. That is one of the things in our patient listening session with the FDA and with the materials that we've shared with them about the patient community that we've tried to express is that we don't think there's going to be one drug that's, going to fix PWS and then we're all going to move on. Some kids, for some kids, the anxiety is the thing. And for some kids, the hunger is the thing. There are going to be, and I personally know, at least one family for which, the Levo drug had side effects, and at least one family for which the DCCR drug had side effects. So I think from a community perspective, from the foundation's perspective, we want as, any drug that is safe and shows efficacy to be approved so that people can work with their doctor and find out what works best for their family. And I think it's reasonable to expect change over the lifetime of an individual with PWS, depending on, what stresses are there. So that's what we're working towards.
This is just a quick plug from another disease population. And my involvement here is very specific to Soleno. So it's only that perspective, but I would just say that the transparency with which, Soleno has guided their communication about their data set can only help with answering these questions. When a company is willing to be that transparent in explaining, their data sets to the community, the physicians and the families, that can only aid in kind of getting to the bottom of those questions.
Thank you, Mindy. I think we've answered most of the questions, if not all of them. We're quite over time as well. So thank you, Susan, for giving us the time, and thank you all to the panelists, and thank you all for listening.
Thank you so much, Anish, for bringing together this fantastic team to talk about DCCR. Having the opportunity to review this with all of you, it really is exciting, and it's time for our community to be excited as well. I want to share my screen just real quick so that you can all see what we have coming up tomorrow. Tomorrow will be our final day of the FPWR conference. We have 4 sessions. I highly encourage all of you. Teresa mentioned it earlier, but join us for the advocacy session tomorrow morning, 11:30 Eastern time. We will be talking specifically about what an advisory committee looks like and how a person can be involved with that advisory committee. It is an opportunity for the entire PWS community to get involved. After that, we'll be talking about strategies for managing anxiety and behavior, as well as feeding techniques for those little ones ages 0 to 2. And we'll end the conference tomorrow talking about sleep disorders and PWS. So please tune in and join us tomorrow and register for those sessions which you would like to have early access to, because we will be making recordings available. Have a wonderful day and goodbye.